My dear friends, welcome to the first most important topic of respiration, and that is glycolysis. Glycolysis is a much more important due to it is a common process for aerobic as well as anaerobic respiration. This is the first process of anaerobic respiration. This is the first process of aerobic respiration. Means this is the common for aerobic as well as anaerobic respiration. Three main scientists were there who had done a lot for this job of glycolysis. Among the first one is Amidon, Gustav Amidon, that is E for Amidon. Second scientist was Meyerhoff, Otto Meyerhoff. So second scientist is Meyerhoff. Third scientist is Panas, Jacob Panas. So third scientist is Panas. By the name of three scientists, Amidon, second Meyerhoff, and third Panas, this entire cycle is nowadays very much famous and known as the EMP pathway. So EMP pathway, E for Amidon, M for Meyerhoff, and P for Panas. Welcome to the pathway EMP, the synonyms of glycolysis. What's happened in glycolysis? Let's go for much more detail on the same. My dear friends, glycolysis is a step which starts from glucose. You all know that glucose is made up of 6 carbon. This is the simple formula of glucose. Here is the first carbon, the second, the third carbon, the fourth, the fifth, and the sixth carbon. Same way, here is the beta D glucose. You know the structure of alpha D and beta D glucose, which has already been studied by you in job of biochemistry. So the first one, this one is a alpha D glucose, this one is a beta D glucose. If you will make a ring formation of this alpha D glucose, it look like this way. If you are talking about beta D glucose, that's look like this way. So OH is on lower side, it is alpha D. If OH is on upper side, it is beta D. OH is on lower side, and over here from today we are taking this alpha D glucose in front of you. So this one is O, it's not a C. So this one is a OH. OH is on lower side, and this is the job of alpha D glucose. Now this alpha D glucose gets works into this entire structure, entire cycle. Let's go towards the actual cycle. In the first step, 6 carbon molecule glucose in presence of hexokinase will be converted into glucose 6-phosphate. Glucose, which is made up of 6 carbon, it will be converted, as I told you, in glucose 6-phosphate. 6-phosphate means on 6th carbon phosphate is there. Please, now in this entire job of glycolysis, there will be too many such type of words. 6-phosphate and 1-phosphate, 3-phosphate, 1,3-biphosphate, 1,6-diphosphate, like that. So please keep in mind, the 6-phosphate doesn't mean that total 6 number of phosphates are there. No, phosphate is only one. Yes, phosphate is only one. But now we can see this is the first carbon, this is the second, this is the third, this is the fourth, here is the fifth. No, that's not fifth. Here is the fifth, and this one is 6th carbon. So on sixth carbon there is a phosphate. So glucose, here it is. Hexokinase is an enzyme. Hexo means glucose is a hexose. Glucose is made up of 6 carbon. So we are telling it as a hexose sugar. So we are now changing this hexose sugar. We are something making change within hexose sugar. So an enzyme name is hexokinase. Each and every enzyme have its own property to be get understood. Over here, the name of enzyme is hexokinase. Now, one should get understand the job that phosphate is added into glucose. Here it is the phosphate, here it is the phosphate. But from where this phosphate came? It should be get think in manner. This phosphate which now just came at glucose, it came from ATP. Yes, here is the adenosine triphosphate. So that phosphate will be get free from ATP and ATP will be converted into ADP plus inorganic phosphate. That inorganic phosphate will be get joined with glucose via name of enzyme hexokinase, and it will be converted into glucose 6-phosphate. So here is the sixth carbon of glucose, and this is the sixth carbon of glucose, and phosphate is added on this sixth carbon. So glucose is being converted into glucose 6-phosphate. So this is the first reaction. Name of enzyme is hexokinase. ATP being get break, phosphate being get free. That phosphate is get joined with glucose. Glucose is converted into glucose 6-phosphate. So via name of enzyme is hexokinase. Now there are total 10 reactions are there. In this window, I am keeping all these reactions step by steps in front of you. In one another window, I will show you each and every single reaction. So now I am showing you the first reaction, the same one, glucose to glucose 6-phosphate in different window. So here it is. Glucose, enzyme is hexokinase, ATP get break, ADP is been get covered by ATP, means ADP been came, ATP is been get break, and that phosphate been get joined with glucose. Please keep in mind on 6th carbon. So phosphate is on 6th carbon. Here it is, phosphate is on 6th carbon. So glucose is converted into glucose 6-phosphate. So this is the first step of glycolysis. Now, my dear friends, before I go further towards all the steps, I want to say you two main phases of glycolysis. The first phase of glycolysis is called as preparatory phase. Preparation is going on. The phase is preparatory phase. Preparation is going on. In this stage, we are not getting ATPs. We are making some preparation to get ATP. I repeat. In this first page, we are not getting ATP. We are making some preparation to get few ACPs. This phase is known as preparatory phase. Over here in this page, we will make a phosphorylation. Means ATP will be used. Yes, actually our aim is to get ATP. But in this case, we are using ATP. So this is the preparation stage. Over here we have used ATP, and by using of one molecule of ATP been get joined with glucose and been converted into glucose 6-phosphate. So that was the first step which has been completed. Let's go towards now second step of this glycolysis reaction. In this second phase, enzyme is phosphohexose isomerase. Now, as I told you, nothing to make a ratto. One has to understand each and every word. This enzyme is been cut with three different words, means been get made by three different words. One is phospho, second one is hexo, third one is isomer ASEAs. Isomer, formation of isomer. You know the main meaning of isomer. So we are making isomer of glucose. What is the isomer of glucose? Fructose. So we are making fructose. But this is the enzyme, that's why we have made the affix ASEAs. So isomer plus ase is equal to isomerase. This isomerase is going on with hexose. So hexose isomerase. Glucose is a hexose, fructose is a hexose. And this isomerase is there not only with hexose but with phospho. So we will say phosphohexoisomerase. By this phosphohexoisomerase, we will convert one hexose phosphate into another hexose phosphate. Means glucose 6-phosphate will be converted into fructose 6-phosphate. Yes, here it is. Glucose 6-phosphate. Via name of enzyme is phosphohexoisomerase. Glucose 6-phosphate is converted into fructose 6-phosphate. This one is of glucose. On 6th carbon, phosphate is there. This one is fructose. This is the first carbon, second, third, fourth, fifth and 6th carbon. This one is 6th carbon. On 6th carbon, there is a phosphate. Again, carbon number 1, second carbon, third carbon, fourth, fifth and 6th carbon. On 6th carbon, there is a phosphate. So on fructose, on 6th carbon, there is a phosphate. Please keep in mind, this is also made up of 6 carbon. So glucose was made up of 6 carbon, glucose 6-phosphate was of 6 carbon, and fructose 6-phosphate is also made up of 6 carbon molecule. So now this much is very much I think so clear in your mind that first of all it was glucose. We have used hexokinase. By that we have converted into glucose 6-phosphate. Now here it is with us glucose 6-phosphate. We are using phospho plus hexo plus isomer plus ase, phosphohexoisomerase, and we are converting into fructose 6-phosphate. So this is the second reaction. We had completed 2 reactions. First was over here, second is this one. So now let's see only second reaction in front of you I am keeping now. So this is the second reaction. Glucose 6-phosphate been converted into fructose 6-phosphate. Reaction number 2, phosphohexoisomerase. It is a glucose 6-phosphate. This one is a fructose 6-phosphate. Carbon number is already written. 1, 2, 3, 4, 5th and 6th carbon of phosphate is there. Again, 1, 2, 3, 4, 5 and 6th carbon of phosphate is there. So this is so simple. Second reaction is completed. Glucose 6-phosphate to fructose 6-phosphates. Let's go towards the third reaction. So now here is the third reaction for you. Up to that reaction we were with fructose 6-phosphate. Please keep in mind everything is made up of 6 carbon. It was with us fructose 6-phosphate. Now we are again using one more ATP. One more ATP will be get break into ADP plus inorganic phosphate, and that phosphate will be get joined with fructose 6-phosphate and will convert into fructose 1,6 biphosphate. Is this made up of 16 phosphate? No, it is made up of 2 phosphate. 2 means biphosphate. Where the phosphates are? Phosphate are at 2 places. Phosphate is on first carbon, phosphates is on 6th carbon. 2 phosphates are there. So now we are telling biphosphate. Let's go towards the name of enzyme. phosphofructose kinase. We are changing from one fructose to another fructose. Yes, phospho is there with fructose. So phospho plus fructose plus kinase. Watch it over here. Let me keep some number of carbon for you. 1, 2, 3, 4, 5 and 6 carbon. We have a phosphate on 6th carbon. Yes, over here. First carbon, second, third, fourth, fifth and sixth. We have phosphate on 6th carbon. Yes, but now we have phosphate not only at 6th carbon, at first carbon also. Previously there was a phosphate. There was a phosphate on 6th carbon. First carbon, second, third, fourth, fifth and 6th carbon. There was a phosphate on 6th carbon. Now there is a phosphate. Now there is a phosphate still on 6 as well as on first carbon. So now there are two phosphate. 2 means biphosphate. On first, on 6, 2 phosphate means biphosphate. So this is the third reaction. Fructose 6-phosphate being converted into fructose 1,6 biphosphate. By adding one molecule of ATP, this fructose 6-phosphate being converted into fructose 1,6 biphosphate. The same reaction in different window. Here it is for you with carbon number. I hope you are now getting everything. Nothing to say. Fructose 6-phosphate, fructose 1,6 diphosphate. Reaction number 3, by use of ATP on 6, there was a phosphate. Still it is on 6. Plus now on first also there is a phosphate. First also there is a phosphate. So that was the third reaction, fructose 1,6 biphosphate. Now my dear friends, this fructose 1,6 biphosphate, it's also made up of 6 carbon. Glucose was of 6, glucose 6-phosphate was made up of 6, fructose 6-phosphate was made up of 6 carbon, fructose 1,6 biphosphate is also made up of 6 carbon. Now this fructose 1,6 diphosphate will be break down into two parts, obviously by name of one enzyme, and the name of enzyme is aldolase. Yes, by use of enzyme, and the name of this enzyme is aldolase. It was made up of 6 carbon. Yes, it was made up of 6 carbon. So now we are 6 carbon sugar phosphate is being converted into 2 molecule of 3 carbon sugar phosphate. 2 molecule of 3 carbon sugar phosphate. So we will make break over here. Yes. Here you can see two different colors are there. At this place we will make a break. And here is your break. Yes, here is your break. Please try to remember something of your previous life. That was something that a fructose 1,6 biphosphate was made up of 6 carbon according to your biochemistry and according to your job of dark reaction. That fructose 1,6 biphosphate, which was made up of 6 carbon, being get converted into 2 molecule of 3 carbon each. Yes, it has been converted into 2 molecules of 3 carbon each. The first molecule of 3 carbon each is glyceraldehyde 3-phosphate. Second one is dihydroxyacetone phosphate. So 2 molecules been get prepared. First is glyceraldehyde 3-phosphate. Second one is dihydroxyacetone phosphate. Remember this glyceraldehyde 3-phosphate. We had told it phosphoglyceraldehyde, PGAL. Glyceraldehyde 3-phosphate means phosphoglyceraldehyde. That was pagal. Dihydroxyacetone phosphate. DHAP. Dihydroxyacetone phosphate. So one material of 6 carbon being get divided into 2 parts, 3 carbon each, PGAL and DHAP. And now my dear friends, here these two are. Yes, so perfect. This one is phosphoglyceraldehyde or glyceraldehyde 3-phosphate, and this one is dihydroxyacetone phosphate, DHAP. One molecule of 6 carbon can give rise to 3 molecule plus 3 molecule of carbon, means 2 molecules of 3 carbon. One molecule of 6 carbon is converted into 2 molecules of 3 carbon each. Fructose 1,6 biphosphate been converted into glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. Enzyme name is aldolase. Aldolase have a function to break one bigger molecule into 2 smaller molecules without using of H2O. Yes, aldolase have a function. Aldolase have a job. Without using job of H2O, it can break one bigger molecule into 2 smaller molecules. So now this is the 4th reaction. Here is the only 4th reaction in front of you to understand in proper way. Fructose 1,6 biphosphate. Enzyme name is aldolase. Been converted into glyceraldehyde 3-phosphate as well as dihydroxyacetone phosphate. Fructose 1,6 biphosphate. 2 parts. One is over here, second one is over here. First one was this one, second one was this one. So this is the 4th reaction. By use of aldolase, we have done it. Now with this, my dear friends, this preparatory phase gets over. Up to this phase we have used ATP. Now we will not use ATP. Now we hope that we will get ATP from the same. Before I go further, now one thing should be keep in mind very properly. I repeat very properly. I repeat very properly. I repeat very properly. Again I repeat very properly, because here we have 2 different things of 3 carbon each. Now from next equations, means next steps, from these 2 we will see only one matter. So what about this second matter? This second matter will be converted into first matter. Something you know, our own story. We have atta to make a chapati, we have atta to make a puri. So in this case we can make that one puri and one chapati. If we want to make only chapati, 2 chapati will be there. So now onwards we will speak 2 molecule of glyceraldehyde 3-phosphate. So now further all reaction will be occurs 2 times. I repeat, this dihydroxyacetone phosphate will be converted into glyceraldehyde 3-phosphates. So all the reactions will be definitely double now always. Like this way, watch it this case. The second phase is called payoff phase. In this entire second phase, watch it what I have done. This glyceraldehyde 3-phosphate is over here. Dihydroxyacetone phosphate is also been get joined in this case. So now onwards we will speak 2 into 3 carbon. So this much part will be 2 times, and next all steps will be 2 times. Further, all steps will be 2 times. I repeat, further, all steps will be 2 times because glyceraldehyde 3-phosphate of 3 carbon will be converted into, sorry, dihydroxyacetone phosphate will be converted into glyceraldehyde 3-phosphate. Means any one will convert into another. But over here, dihydroxyacetone phosphate, DHAP, is going to get converted into PGL. So we will get 2 molecules of PGAL. So let me enlarge the same for you in proper way. Yes, now watch it the same. You can see the same now perfectly. This is glyceraldehyde 3-phosphate. Here it is. This one is also glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate is been converted into glyceraldehyde 3-phosphate. So now we will write like this way: 2 into 3 carbon. So 2 molecule of glyceraldehyde 3-phosphate. Yes, it is glyceraldehyde 3-phosphate. This one is spelling wrong somewhere. This one is glyceraldehyde 3-phosphate. Now, my dear friends, please keep one more thing in mind. Up to this one was a differ phase. Now it is payoff phase. Means in this page we will get ATP. We will get NADH. These two will be again we will use. Be with me now. Name of enzyme is, this one is made up of 3 carbon. They both are with phosphate. Dihydroxyacetone phosphate been converted into glyceraldehyde 3-phosphate. So DHAP is of 3 carbon, been converted into glyceraldehyde 3-phosphate of 3 carbon. So these both are the isomer of each other. Both are made up of 3 carbons, so triose. Both are with phosphate, so phosphate. Both are the isomer of each other. Isomer is going to be get formed by this enzyme, so enzyme have affix ASEase. Isomerase. Triose, phosphate, isomerase. Don't do it like your friends. Don't make anything recto. Don't make anything recto. 3 carbon, so triose. Phosphate is there, so phospho. They both are isomer of each other, so isomer. That is the name of enzyme, so ASEase. Triose, phosphate, isomerase. In one line, dihydroxyacetone phosphate has been converted into glyceraldehyde 3-phosphate. So now we have 2 molecules of glyceraldehyde 3-phosphate. Yes. Step number 5. I hope you got now everything. Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, 2 molecules of glyceraldehyde 3-phosphate. Triose, phosphate, isomer, ASEase, triose, phosphate, isomerase. So this was the 5th step. Now I am moving towards the next step over here in front of you. So now let's move towards step number 6. We have with us glyceraldehyde 3-phosphate. By the word dehydrogenase enzyme, we are making dehydrogenation. D means delete. Delete of hydrogenation. Hydrogen is going to be get removed. So glyceraldehyde 3-phosphate enzyme is glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate plus dehydrogenase. Dehydrogenation is going on and we are getting 1,3-biphosphoglycerate. So phosphate is on 1st carbon, phosphate is on 3rd carbon. Here we were with 3-phosphate, is it? No. We were with 1-phosphate on 3rd carbon. Now we are with 13-phosphate, is it? No. We are with 2-phosphate on 1st and 3rd carbon. So we were with 1-phosphate, now we are with 2-phosphate. We were with 1-phosphate on 3rd carbon. We were with 1-phosphate on 3rd carbon. Now we have 2-phosphate on third as well as on first, so we are speaking 1,3-biphosphate. Let's see the number of carbon. First carbon, second carbon, third carbon. And we have a phosphate on third carbon. Same way. First carbon, second carbon, third carbon. We have a phosphate on third carbon as well as we have phosphate on first carbon. So one phosphate is added. And then from where this phosphate came, my dear friends? From ATP? No. First time it is came from H3PO4. And yes, hydrogen is removed. So who will come over here to get this hydrogen? Obviously, hydrogen acceptors. And here is the reaction. Yes. And this reaction, 2 molecules of phosphate will come, 2 NAD will come, 2 NAD will be converted into 2 NADH2 and 2H plus will be get free. 2 times we are removing H2. Why 2 times? We have 2 molecules. Why 2 molecules? I taught you too many times. This has been converted into that. So now together we can get understand, as I told you too many times, that this reaction will occur 2 times 2 molecule of 3 carbon. 2 molecule of 3 carbon. So we'll use 2 phosphate, we'll use 2 ADP, we'll get 2 NADPH, we'll get 2H plus. Yes, 1,3-biphosphoglycerate means 1,3-biphosphoglyceric acid in different word. Yes, 1,3-by-PGA. If anyone among you remember something, please try to remember photosynthesis. Photosynthesis was a job of reduction to make food, to make glucose. This is the glycolysis. This is a job of oxidation to break glucose. Remember the dark reaction of photosynthesis. It was definitely 100% vice versa of this reaction. Try to catch this much after this end of this session. It was, definitely yes, it was. Now let me keep myself on this 6 point. Glyceraldehyde 3-phosphate, 2 molecules, will be converted into 1,3-biphosphoglyceric acid, 2 molecules. Over here we are removing hydrogen. So name of enzyme is dehydrogenase. Dehydrogenation means oxidation. Photosynthesis was the job of reduction. This is respiration. This is the job of oxidation. As well as we are adding phosphate. So phosphorylation. Oxidation and phosphorylation. Both the things are going on 6th process. Now let us see the 6th process in different window. The only 6th process. Yes, here it is. Glyceraldehyde 2-phosphate been converted into 1,3-biphosphoglyceric acid. Phosphate was there on carbon number 1, no. On 2, no. On 3, yeah, it was on third carbon. Now phosphate is on first carbon. This one is the first carbon and this one is third carbon. Phosphate is on third carbon also. So 1,3-biphosphoglyceric acid. We were with 2 molecules, so 2 phosphate been added, 2 NAD came, 2 NADH came out, 2H plus, 2 times H plus, 2 times H plus came out. So this is the 6th job. and this is been get completed. We got 1,3-biphosphoglycerate. Now onwards I am showing you the seventh step. So let's go towards seventh step. Here it is for you. Sixth step been get completed. Now we are on seventh step. 1,3-biphosphoglycerate been converted into 3-phosphoglycerate. What means? There were 13 phosphate? No. On first carbon, on third carbon, 2 phosphates were there. Now are there 3 phosphate? No. On third carbon, only 1 phosphate is there. Yes. On first, on third, 2 phosphates were there. Now only third, 1 phosphate is there. From first phosphate is gone. From first carbon, phosphate is gone. Where is phosphate gone from first carbon, my dear friends? Will be here in front of you. Yes, here it is. Perfect. ADP will come, will get this phosphate and will convert into adenosine triphosphate. Here, my dear friends, we got 2 ATPs. Why? So simple. We have 2 molecules, as I told you to remember in perfect way. We have 2 molecules of glyceraldehyde 3-phosphate. Remember again, dihydroxyacetone phosphate was converted into glyceraldehyde 3-phosphate. So the process is going on 2 times. This entire process will run for 2 times. So again, on first carbon, on third carbon, on first, on third, on both the carbon, phosphate was there, biphosphate. Now on third carbon, phosphate is there, but on first carbon there is no phosphate. So where this phosphate gone from this first carbon? So perfect. The phosphate from first carbon went to join with ADP to form ATP. Means first time we got ATP in this process. First ATP formation. That ATP is going on in this cycle. So this ATP formation is known as substrate level ATP formation. Substrate level phosphorylation. Phosphorylation means what? Means I told you earlier, ADP plus inorganic phosphate fused together to form ATP. That is the job of phosphorylation. Over here also phosphorylation is going on. But this phosphorylation is not going on in electron transport system. Was that? We'll discuss it in further lectures. But this phosphorylation is going on during this cycle, during this phase, during this process, and that is why it is called as what? That is why it is called as substrate level ATP formation, means substrate level phosphorylation. So now let's go towards the seventh point only to get in proper way. So here it is the only seventh point. Nothing more to say. On first carbon phosphate was there, on third carbon phosphate was there, now only on third carbon phosphate is there. So 1 plus 3 biphosphate been converted into 3 phosphate. This occurs with 2 molecules. Yes. So 2 ADP will be get converted into 2 ATP. So up to this we have completed 7 point. Yes, out of perfect 10, 7 is completed. Here that perfect 10 is there. Let's go towards point number 8. Here it is for you. 3-phosphoglycerate, it was there with us. Now it is converted into 2-phosphoglycerate. So we will get ATP over here, is it? It should be. Why it should be? Because of there were 3 phosphate. Now there are 2 phosphate. Is it look like that? No, not at all. We will not get any ATP over here. Phosphate was there on third carbon. So we have written 3-phosphate. Now phosphate is on second carbon. So we are writing 2-phosphate. So only position of phosphate from third carbon to been shifted to second carbon. So mutation is occurred, position change. In genetics it is called mutation. Something changed, something different came. Mutation is occurred. So in genetics we are speaking word mutation. So now by that word we are using mutase. Enzyme name is mutase. Phosphoglycerate mutate. Phosphoglycerate was there. Mutation been occur with phosphoglycerate. So phosphoglycerate mutate. So simple, mutation been occur with phosphoglycerate. So what happened? On third carbon there was a phosphate. Now this phosphate is on second carbon. Here is the only reaction number 8 for you. On third carbon there was phosphate. Now on second carbon there is phosphate. Total 2 molecules were there. Total 2 molecules were there. Will we get ATP? No, not at all, because we are not removing any phosphate, we are not adding any phosphate, we have only changed the site of phosphate, we have only changed the place of phosphate. So that is the point number 8 which we have completed. 2-phosphoglycerate. Over to step number 9. I am keeping in front of you. This is step number 9 now for you. Here it is. My dear friends, in this 9th step, yes, in this 9th step, you can see now the matter is 2-phosphoglycerate been converted into 2-phosphoenol pyruvate. Yes, PEP, phosphoenol pyruvate. So what the difference over here? Only we are removing one molecule of H2O. Now if you will say, sir, over here 2 molecules written and you are telling 1 molecule. So my dear friends, as I told you too many times, this entire reaction is going on 2 times because this DHAP been converted into, yes, phosphoglyceraldehyde. So this process done 2 times because we have with 2 molecules we are getting 2 molecules, so we will get 2 molecules. So now what happened in this process? H2O will be moved out. H gone, H gone, O gone, H gone, H gone, O gone. H2O is gone from this process. So by removing H2O by using enzyme named by enolase in this point number 9, from 2 molecules of phosphoglycerate we will get 2 molecules of phosphoenol pyruvate. 2 molecules. Name of molecule is 2-phosphoglycerate. 2-phosphoglycerate means 2-phosphate? No, again. Phosphate was on second place. So let's see the thing in different window. 2-phosphoglycerate. Second position, phosphate is there, 2-phosphoglycerate. H2O is removed out. Been converted into phosphoenol pyruvate, PEP. So this is the phosphoenol pyruvate. We were with 2 molecules. We got 2 molecules. So obviously 2 H2O will come. We were with 2 molecules. We got 2 molecules. Obviously we will get 2 molecules of H2O. ये जो ब्रैकेट में 2 लिखा है ना मेरे दोस्तों, that is for 2 molecules. This is written for 2 molecules. This 2 is for what? 2-phosphate? No, 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 no. This 2 is for phosphate is on second position. By using enolase, 2-phosphoglycerate been converted into phosphoenol pyruvate, point number 9. And now the final step, that will be the 10th point, which I'm keeping in front of you now for you. So my dear friends, phosphoenol pyruvate was made up of 3 carbon. Still it has been converted now with another molecule of 3 carbon, and that is pyruvate. The enzyme is pyruvate plus kinase, changing in pyruvate, pyruvate plus kinase. Now try to get one word. Over here it was written phospho. This one is only pyruvate. This one is written phosphoenol pyruvate. Pyruvate is here. Phosphate is removed. Yes. And if phosphate is removed, now we will get ATP over here. So this is entire Payoff phase. This is entire second phase. Point number 10. Phosphoenol pyruvate been converted into pyruvate. I repeat, pyruvate was there, pyruvate is there. Pyruvate was there, pyruvate is there, only the phosphate is been get moved out. So that phosphate will be get joined with ADP and we get ATP. Why we get 2? So perfect, because we were with 2 molecule and we are getting 2 molecule. That we have discussed a lot. So this is the 10th job together, everything. So this much is completed for you perfectly. Point number 10, and that is phosphoenol pyruvate been converted into pyruvate. Point number 10. In 10th point, again we are getting ATP, so it's a second ATP formation. So my dear and near friends, in this Payoff phase, we got 2 times ATP. We got 1 ATP over here. We got another ATP over here. Both the times we got 2 ATPs. So how many ATPs we got? 4. How many ATP we have loosed before? 2. Yes, we have loosed before 2 ATPs. Let me show it to you for you. Here it is. We have loosed 2 ATP in preparatory phase. So in preparatory phase we lose 2 ATPs. In Payoff phase we got 2 ATPs. In preparatory, first ATP loosed. In preparatory, second ATP loosed. Over here we got first time 2 ATP. Over here we got second time 2 ATP. So we got total 4 ATPs, and we lose 2 ATP. So net we are with profit of 2 ATPs by the end of this process. Yes, so this was the exact reaction, the 10th one and the final one, and this is phosphoenol pyruvate been converted into pyruvate, and I am showing you the only 10th reaction. Here it is for you, only 10th one. Phosphoenol pyruvate been converted into pyruvate. This is phosphoenol pyruvate and this is pyruvate. We were 2 molecules of phosphoenol pyruvate. We are with 2 molecules of phosphoenol pyruvate. And if we were 2 molecules, so we are getting 2 ATPs. So with this point number 10, the final cycle gets end. Here is the final ends part of cycle. I again repeat, in the second half, we are getting 2 times ATP. We are getting 2 times ATP, and that is 2 ATP every time. We are getting 2 times ATP, and that is 2 ATP every time. So how many ATP we got? 4. In previous stage, in starting in first half, we lose 2 ATP. So 4 minus 2, net profit is of 2 ATP. So this was all for the job of glycolysis. By name of scientist, it is also called as EMP pathway. Too many different names are there of glycolysis. We'll talk it in general, another matter, another part, another video. So thank you very much. Jai Hind.